Dear students, in the last lecture, we have studied in detail the de novo synthesis of purine ribonucleotides and the salvage pathways for purine biosynthesis. We have also seen in detail the symptoms, causes, and treatment of Lesch-Nyhan syndrome. So in today's lecture, we are going to see degradation of purine ribonucleotides, synthesis of deoxyribonucleotides, and disorders associated with purine catabolism. So today's learning objectives are focused on studying synthesis of deoxyribonucleotides, then catabolism or degradation of purines, and then we'll see in detail disorders associated with purine degradation. For example, adenosine deaminase deficiency, purine nucleoside phosphorylase deficiency, and gout. We are going to start a very important topic of nucleotide metabolism, and that is synthesis of deoxyribonucleotides. In the previous lecture, we have studied that how can we produce purine ribonucleotides in the form of purine ribonucleoside monophosphates. For example, adenosine monophosphate and guanosine monophosphate. These two nucleotides are found in RNA. But when we have to make nucleotides for DNA, there is a need to convert ribonucleotides into deoxyribonucleotides. Now for this conversion, we need enzyme, and that enzyme is called a ribonucleotide reductase or RR. This enzyme is crucial for making deoxyribonucleotides from ribonucleotides. But there is one condition for ribonucleotide reductase to work. Ribonucleotide reductase, it can only work when ribonucleotides are present in ribonucleoside diphosphate form. For example, adenosine diphosphate and guanosine diphosphate and others. So every ribonucleotide which has to be converted into deoxyribonucleotide must be present in its diphosphate form for the action of ribonucleotide reductase enzyme on it. So we need to convert all ribonucleoside monophosphates, for example AMP and GMP and others, first into ribonucleoside diphosphate, for example ADP, GDP and others. So then only ribonucleotide reductase would work on them and will make for us deoxyribonucleoside diphosphates. Now the same enzyme, that is ribonucleotide reductase, can work on pyrimidine ribonucleotides as well. So this enzyme is single and general, and it can work on both purine ribonucleotides and pyrimidine ribonucleotides and convert them into deoxyribonucleotides for DNA. This figure is explaining conversion of ribonucleotides to deoxyribonucleotides by ribonucleotide reductase enzyme. So you need always ribonucleoside diphosphate whenever you have to create deoxyribonucleoside diphosphate. For example, if you have AMP and you want to make deoxy AMP from it, then first you have to convert AMP into ADP. Now what is ADP? ADP is ribonucleoside diphosphate. Now this ADP will be converted into DADP with the help of ribonucleotide reductase enzyme, and then this DADP will be removing its phosphate group and then you can convert it into DAMP. So this is the whole series of reaction which is followed for making AMP into DAMP. So the requirement for ribonucleotide reductase is this, that ribonucleotides must always be present in their diphosphate forms. So here is ribonucleoside diphosphate, which condenses with thioredoxin. It is basically a coenzyme for ribonucleotide reductase, and this is present in its reduced form. As you can see here, the two hydrogen atoms with the thiol group. So these hydrogen atoms of thioredoxin can be donated to ribonucleoside diphosphate, and it can become reduced to deoxyribonucleoside diphosphate. Basically, ribose sugar is converted into deoxyribose sugar. Now in this reaction, thioredoxin itself is oxidized to thioredoxin into its thioredoxin oxidized form, which is here represented by double S. There is no hydrogen atom present in it. And water is also released in this reaction. Now this oxidized thioredoxin may be converted back to its reduced form by taking hydrogens again from NADPH. NADPH, most important source, as you know, is HMP pathway. So NADPH itself becomes oxidized, but thioredoxin becomes reduced to recycle the same process. And the enzyme which is required here is called thioredoxin reductase. Now, ribonucleotide reductase enzyme can be inhibited by high levels of DATP. If deoxyadenosine triphosphate is present excessively in the body, then it can inhibit ribonucleotide reductase, and thus no more deoxyribonucleotides will be produced in the cell. Now we see the regulation of ribonucleotide reductase enzyme. Ribonucleotide reductase contains two important subunits. One is R1 subunit, another one is R2 subunit. R1 subunit has two sites. One site is called substrate specificity site, and this site is called activity site. These two sites are also called as allosteric sites or regulatory sites. While R2 subunit, it has got catalytic site where the actual reaction takes place. Okay, so activity site can control the activity of overall enzyme. For example, if with the activity site ATP is bound, then the whole enzyme would be activated. But if the activity site is bound with DATP, that is deoxyadenosine triphosphate, then the whole enzyme will be inhibited. Substrate specificity sites can bind to different substrates, and one substrate, when bound, can control the synthesis of other substrate. So the substrate that are specific for substrate specificity sites are ATP, DATP, DTTP, or DGTP. For example, if DTTP is bound to substrate specificity site, then it will be making the other nucleotides, for example, DGDP. So DTTP, if bound to the substrate specificity site, then it will be giving the message to ribonucleotide reductase to make DGDP. So one substrate specific for substrate. So one substrate which is specific for one substrate specificity site can control the formation of other nucleotide. And R2 subunit is the catalytic site where the actual reaction takes place, and ribonucleoside diphosphate is converted into deoxyribonucleoside diphosphate. Now, degradation or breakdown or catabolism of purines. So this protocol is for digestion of dietary nucleic acids. So RNA or DNA in the mouth remain unaffected, but when they arrive in the stomach, there is low pH, which can denature nucleic acids, and you can get denatured nucleic acids in the stomach. Then these are poured into duodenum. In the duodenum, there are pancreatic secretions, for example, nucleases and phosphodiesterases. Nucleases first convert denatured nucleic acids into oligonucleotides, and then by the action of phosphodiesterase, we get mononucleotides. Then the enzymes from the small intestine, for example, nucleotidases, convert mononucleotides into nucleosides by eliminating phosphate group from them. And then again, another set of enzymes called nucleosidases from small intestine convert nucleosides into pyrimidines and purines, and their sugars are also released from them. Sugars, whether deoxyribose or simply ribose, are well absorbed by this circulatory system, while pyrimidine and purines are not well used up, and these are converted into uric acid and then get eliminated from the body via urine. Okay, this slide is depicting the endogenous breakdown of nucleotides into uric acid. So if you have adenosine monophosphate, if you have inosine monophosphate, or even if you have guanosine monophosphate, all of these purine nucleotides are end up into uric acid. So we are going to start from PRPP, which is the precursor for nucleotides, and that is phosphoribosyl pyrophosphate. It is converted into 5-phosphoribosylamine by using nitrogen in the form of amino group from glutamine. And here the enzyme used is glutamine phosphoribosyl pyrophosphate amidotransferase. And then after going number of steps, we get this parent purine nucleotide, which is called inosine monophosphate. Now this inosine monophosphate can be converted into inosine by nucleotidase enzyme. In this reaction, water enters and inorganic phosphate leaves the reaction. Now, if you have AMP, then AMP with the help of AMP deaminase is converted also into inosine monophosphate, which can then further be converted into inosine. And this enzyme basically replaces amino group from AMP by water. AMP can also be converted into adenosine by nucleotidase enzyme. Here water replaces its phosphate group. This adenosine with the help of adenosine deaminase is also converted into inosine. And here amino group is replaced by water. So from AMP or IMP or adenosine, one intermediate is generated in the degradation of purines, and that is inosine. Now this inosine is converted into hypoxanthine by using purine nucleoside phosphorylase, which is also abbreviated as PNP. In this reaction, phosphate group enters the reaction while ribose 1-phosphate, that is the sugar, it leaves the reaction, and we are just left with the base, which is called hypoxanthine. This hypoxanthine undergoes oxidation in the presence of xanthine oxidase enzyme. Hydrogen peroxide is released as a side product, and hypoxanthine is eventually converted into xanthine. This xanthine again oxidizes in the presence of xanthine oxidase, and we get ultimately uric acid. If you have guanosine monophosphate nucleotide, then it will be first converted into guanosine with the help of nucleotidase enzyme, and then guanosine will be converted into guanine base with the help of purine nucleoside phosphorylase or PNP enzyme. Here again, phosphate group is going to be entering in the reaction, and ribose 1-phosphate would be leaving the reaction. Now guanine is going to be converted into xanthine with the help of guanase enzyme. Here water enters the reaction and amino group leaves in the form of ammonia. Now this xanthine is oxidized with the help of xanthine oxidase, finally to uric acid. So if you have AMP, if you have IMP, or if you have GMP, all of these nucleotides are ultimately converted into uric acid. Disorders, defects, or diseases associated with purine catabolism. We are going to do three disorders, namely ADA deficiency, PNP deficiency, and gout. Now in ADA deficiency, definitely there is deficiency of ADA enzyme, which stands for adenosine deaminase enzyme. This enzyme is found in high concentrations in lymphocytes in human beings. So if this enzyme is deficient, then lymphocytes are mostly targeted and they may be depleted. If adenosine deaminase enzyme is deficient, then there is high accumulation of adenosine. So adenosine becomes elevated, and this adenosine may be converted into ribonucleotides, or it might also be converted into DATP. When DATP rises, then it may inhibit enzyme ribonucleotide reductase. When ribonucleotide reductase enzyme becomes inhibited, then deoxyribonucleotides cannot be formed. When deoxyribonucleotides cannot be formed, then DNA synthesis is impaired. And when the DNA synthesis, this is affected, then there is depletion of lymphocytes causing lymphocytopenia. And this is due to developmental arrest. And apoptosis of lymphocytes. There is programmed cell death of lymphocytes, and ultimately leading to lymphocytopenia. Now the lymphocytes that may be depleted in this condition may involve T cells, B cells, or natural killer cells, and ultimately this will be leading to severe combined immunodeficiency disease, or SCID. If there are children who are having ADA deficiency and if they remain untreated, then they can die before 2 years of age. The only treatment available for this type of enzyme deficiency is bone marrow transplantation, enzyme replacement therapy, or gene therapy. Now the second disorder associated with purine degradation is purine nucleoside phosphorylase deficiency, or PNP deficiency. This enzyme may be missing and inosine cannot be converted into hypoxanthine, and therefore the whole sequence, which is afterwards hypoxanthine, will be inhibited. Like that of ADA deficiency, purine nucleoside phosphorylase deficiency is also autosomal recessive, but its prevalence is rare as compared to ADA deficiency, and this is also less severe than adenosine deaminase deficiency since it can only affect T cells. Patients of PNP deficiency encounter recurrent infections and also neurodevelopmental delays. Now the third important disorder associated with purine catabolism is gout. Gout is basically a complication of hyperuricemia. If high levels of uric acid in the blood become persistent, then it may be precipitated into gout. So the main reason behind the prevalence of gout is the overproduction of uric acid or either under excretion of uric acid. Maybe uric acid is formed in massive concentrations in the body, or maybe it is found in the normal concentrations but cannot be excreted well by the kidneys. Types of gout. There are two types of gout, renal gout and metabolic gout. But the most commonest cause of gout is renal gout, and renal gout is due to under excretion of uric acid, which indicates that kidneys become unable to clear uric acid from the body. There are two further types of renal gout. First is primary renal gout, another is secondary renal gout. Primary renal gout is due to some idiopathic reasons. For example, some unidentified genetic excretory defects may lead to under excretion of uric acid. while secondary renal gout is associated with those diseases which affect the kidney normal process. For example, if a person is having lactic acidosis, then that person may develop hyperuricemia. What is the reason behind it? That lactic acid and uric acid compete for the same kidney transporter for excretion from the body. So if there is more lactic acidosis, then the person is at more risk for developing hyperuricemia. The second main reason behind the prevalence of secondary renal gout is the association of environmental factors. For example, if a person on drugs, for example thiazide diuretics, so these drugs longer consumption can damage kidney, and as a result of which renal gout may be precipitated. Similarly, if a person is exposed to lead, then lead can also result into damage to kidney, and it may also lead to renal gout. Now the gout which is associated with lead is also called as satanin gout. The second most important cause of gout is metabolic gout, which is associated with overproduction or oversynthesis of uric acid in the body, but this is considered as less common cause of gout. It is subdivided into primary metabolic gout and secondary metabolic gout. Now, primary metabolic gout is mostly idiopathic, which means that the reason or the cause is unknown, but several mutations have been documented in the gene of phosphoribosyl pyrophosphate synthetase that may lead to hyperuricemia. As we know that PRPP is a precursor for de novo synthesis of purines. So as more purines are formed, there is more degradation of purines. So as a result of which hyperuricemia can be observed in the patients with PRPP synthetase mutations. Another important cause of primary metabolic gout is Lesch-Nyhan syndrome, about which we have seen in detail in our last lecture. Secondary metabolic gout is mostly due to when purines are available in increased amounts. For example, the patients with myeloproliferative disorders like leukemias or cancers or tumors, or even those patients who are undergoing chemotherapeutic trials. So these patients have also high rate of cell turnover, which may result into increased availability of purines. So more purines available, more purines degradation, more uric acid in blood. It has also been seen that hyperuricemia may be associated with some metabolic diseases which are definitely unrelated, but these may result into hyperuricemia. For example, we have seen in glycogen storage disease, von Gurki disease, and we have seen in detail that how von Gurki disease can be linked to hyperuricemia. So you can recall that lecture. Similarly, in the metabolism of fructose and galactose, we have also studied in detail fructose intolerance and its association with hyperuricemia. Clinical manifestations of gout. Patients of gout can have deposition of uric acid in the form of monosodium urate crystals in their joints, and as a result of which an inflammatory response is triggered. And it may cause first acute gouty arthritis, and then it can progress or lead to the development of chronic gouty arthritis. Gout patients may also have some nodular masses of monosodium urate crystals, which are called as tophi, and these may be seen in the soft tissues of the body, resulting into chronic tophaceous gout. For example, here you can see the presence of tophi on the index finger of gout patient. In the patients of gout, even uric acid stones in the kidney may also be seen, and this condition is called urolithasis. Now, how can we make diagnosis of gout? This is mostly done through a technique which is called as arthrocentesis. So this is basically a procedure through which synovial fluid from an affected joint of a person with gout is aspirated and then it is examined with polarized light microscopic technique in order to confirm the detection of a yellow colored needle-shaped crystals of monosodium uric acid. For example, here you can see this technique arthrosynthesis, by which synovial fluid is aspirated and then examined with the help of microscopy, polarized light microscopy. And here are the crystals which are yellow colored, needle-like, and these crystals are basically of monosodium uric acid. Now, what treatment options we have to fix gout? Since gout is an inflammatory disease, so acute attacks of gout can be treated well with anti-inflammatory drugs. For example, colchicine or some steroidal drugs such as prednisone, and non-steroidal drugs, for example, indomethacin. For the patients of renal gout who are under excretors of uric acid, can be administered with uricosuric agents or drugs, for instance, probenecid or sulfenpyrazone. These are the drugs that target kidneys and help increase the excretion of uric acid from the body. For the patients of metabolic gout, which is associated with overproduction of uric acid, a drug of choice is allopurinol that can be given to these patients. Allopurinol is a structural analog of hypoxanthine, and it inhibits or blocks uric acid synthesis, and thus this is used to treat metabolic gout. For your MCQ part, you must know that allopurinol is converted first in the body to another compound which is called oxypurinol. This oxypurinol basically acts as an inhibitor of xanthine oxidase enzyme. When this enzyme is inhibited, it results into an accumulation of two other products, that is hypoxanthine and xanthine. But hypoxanthine and xanthine are more soluble than uric acid, and therefore these are less likely to trigger inflammatory responses in the body. There is another drug which is currently used for the treatment of gout, and that is febuxostat. It is also sold with the brand name Uloric. It is basically an inhibitor of xanthine oxidase, but its mode of action is different from the other drugs. It basically inhibits molybdenum site on the xanthine oxidase, which is the active site of xanthine oxidase, and thus it blocks the action of xanthine oxidase and used to treat hyperuricemia and ultimately gout. Okay, students, activity for you. You are supposed to recall today's lecture and give the answers of these two queries. I'm going to reveal the answers. If you have answered both queries appropriately, it means you have understood the all concepts of today's lecture.